Avian influenza virus, one of the most important viruses, and it has a zoonotic importance. Avian influenza virus is a problem for poultry around the world. The virus is unusual in that it can cause a range of disease symptoms, from causing a subclinical infection to being highly virulent with 70% more lesions appear as necrosis of the comb and wattle. Symptoms appear as necrosis of comb and wattles and apathy of the birds. There is some lesions in the proventriculus, hemorrhages in the mucosa of the proventriculus, subcutaneous necrosis of foot bags, conjunctivitis and edema of the head, nerve signs. The main reservoir for the virus is in wildlife, therefore complete eradication is not possible. It's very difficult. Also, influenza, because of its broad host range, can represent a zoonotic risk. All these factors make avian influenza an important but difficult pathogen to control in our poultry population. Influenza virus are negative sense segment RNA virus in the family Orthomyxoviridae. Influenza virus can be subdivided into three different antigenic types, A, B, and C. However, only type A influenza virus are of veterinary importance, since type B and C influenza virus are human pathogen. Type A influenza virus have 8 different gene segments that encode 10 different viral proteins. They can be divided into the surface glycoprotein, the heme agglutinin, HA, and the neuraminidase NA proteins. The heme agglutinin and neuraminidase gene also have the most sequence and antigenic variation of all these influenza proteins. The heme agglutinin protein has 16 defined antigenic subtypes, H1 to H16, and the NA has 9 antigenic subtypes from N1 to N3. The nomenclature for describing influenza virus has been standardized to be consistent for influenza virus. The feature used to describe all new influenza virus are antigenic type A and B and C, host animal which is the virus was isolated from, geographic location which the isolate came from. This can be city, state, provenance, or country designation, laboratory or other reference ID numbers. Since a single lab often makes multiple isolation in a year, a unique identifier number is often included for each isolation. The year of the isolation. The heme agglutinin and neuraminidase subtypes are often included in the parentheses at the end. For example, the Turkey influenza virus in 1999 from Missouri would be A, type A, influenza type A, Turkey. This is the species. Missouri, this is the locations. This is the reference number, and this is the year of isolation. And H1 and 2, this is the neuraminidase and heme agglutinin protein. The virus has caused localized infection are usually referred as low pathogenic avian influenza virus, and typically these viruses don't cause high mortality in affected flocks. The virus that cause systemic infections usually cause high mortality and are referred to as high pathogenic avian influenza virus. The high pathogenic avian influenza virus for unknown reason are restricted to the H5 and the virus are low pathogenic. Influenza virus have a wide host range and routinely infect humans, swine, horses, chicken, turkeys, and wild birds. Although influenza virus can become endemic in all these populations, the natural host and the reservoir for the virus is in the wild ducks, gulls, and sherbirds. Clinical signs and lesions. The disease lesion with avian influenza in poultry can be extremely variable depending on the viral isolate, species of the infected bird. Other factors include the presence of other pathogens, immune status of the host, age of the bird, environmental factors. In general, for low pathogenic avian influenza virus, the disease symptoms are restricted to the respiratory tract and can include lesion in the sinuses, trachea, bronchi, lungs, air sacs, intestine. Decrease in egg production without other clinical signs are common in layer or breeder infections. The high pathogenic avian influenza virus H5N1, there is some lesions, congested hemorrhagic pneumonia with blood clots in the thoracoabdomen, without associated traumatic lesion. High pathogenic avian influenza virus, hemorrhagic suffusions in the heart. High pathogenic avian influenza virus H5N1, pancreatitis with necrosis, pancreas between the two lobes of the duodenum. Pathogenic avian influenza virus, hemorrhage of the ovaries